right. Well, I'm very happy to be able to introduce this next section of our training. This one is going to be focused on using comparative genomics for viral molecular epidemiology. So I thought I would take a little bit of a step back and talk about some of the underlying concepts that are needed to interpret a phylogenetic tree and lay the foundation, hopefully, for the remaining sections. So my title today, Comparative Genomics for Public Health, and specifically interpreting phylogenetic trees. There we go. Is this one? Okay. So when we take a virus, sample it and sequence it, and we have a population of viruses that we've sequenced over a epidemic, we are examining a snapshot of the genetic diversity. Evolutionary processes are what give rise to genetic diversity. So there are four basic mechanisms that we need to actually consider. Those are mutation, migration, genetic drift, and natural selection. And these different processes will either increase or decrease the variant frequency that we might see. So if we have selection for a variant that can outcompete its other variants that are circulating by replicating faster within a host, the other variants are going to die off. So natural selection is going to reduce those other variants and select for that one variant that is successful and can outcompete its others. We often analyze genetic data by building phylogenetic trees. And when we do sequence that population of viruses, the genetic diversity is a result of those evolutionary processes. So when we build phylogenetic trees, this is the definition that's in Wikipedia. Wikipedia says a phylogenetic tree or an evolutionary tree is a branching diagram of evolutionary relationships based upon similarities and differences. This is a unsatisfying definition as a scientist. I am not an artist. I don't do diagrams. I test hypotheses and generate hypotheses and investigate phenomenon to understand the truth around me and what may have given rise to those processes. So the definition that I use is that a phylogenetic tree is an evolutionary hypothesis. It is a hypothesis that allows us to understand some of those evolutionary processes, and it shows the history and relationships through a process of common descent of the observed genetic diversity. And importantly, when I say common descent here, is that I believe evolution can be observed in a tree-like pattern. We know that this is not 100% true. We know that we have a phenomenon like recombination and lateral gene transfer that might mean that evolution is more like a banyan tree with many different trunks than just a classical view of it being just a regular tree. So this drawing is one of the first drawings of an evolutionary tree, and one that describes the relationships. And it was a hand-drawn note in one of Darwin's notebooks. So the theory of evolution is often given credit to Charles Darwin and Alfred Russel Wallace. They formulated the theory of evolution by common descent, and Charles Darwin gains a lot more credit for it from his publication of The Origin of Species in 1859. In 1920 and 1940, we had a modern synthesis that brought together concepts of ecology, population genetics into an evolutionary framework. So that modern synthesis, again, 1920 and 1940, this is all prior to knowing what DNA was, and that the gene itself was the unit of evolution. But between that period of 1950 and 1980, we went through a rapid discovery phase where we characterized the molecular clock, the neutral theory of evolution, and developed methods for comparative genomics. Ecology proposes mechanisms for genetic variation between populations, Epidemiology studies the patterns of variation or patterns that cause disease and the effects of diseases within populations. And an ecological study could be considered an epidemiological study in which the unit of analysis is a population, not an individual. Okay, so ecology is interested in populations, epidemiology, what happens between populations and how things interact. Epidemiology is interested in what's happening within populations, and an ecology study is really an epidemiology study where we're not using units as being individuals, but whole populations. And I think this is important to understand because this allows us to bring ecological concepts into epidemiology. That we're really just the difference, the fundamental difference is the scale. We're not talking about sick people. We're talking about virus. We're not talking about viruses moving between individuals. We are talking about how viruses move between populations. In the last 10 years, which really brings us to this point, is that we've had this massive amount of sequence data. The previous section allowed us to examine how some of that data is being generated and the new methodologies that are coming forward. We've also had increased computational power. My cell phone is more powerful than the computer I use to do my PhD. And then we have theoretical and analytical advances that have really allowed us to start unifying epidemiology, ecology with evolution in order to really bring this field of molecular epidemiology of viral infections to its current state. In 2004, this term phylogenomics was introduced. This was introduced by Brian Grenfell in a paper in Science, and I'm quite happy to share this with you. This is one of the most important papers that I've read in my life, and it gives some great examples of how to interpret phylogenetic trees in an epidemiological framework. And they describe how genetic variation is affected by host immunity, spatial transmission, and those epidemic dynamics. So the faster a virus moves through a population, the more people that are infected, the greater the amount of genetic variation that you will see. As viruses move between different locations and become, if a virus is circulating here in Georgia and another virus is circulating in Melbourne, Australia, those viruses are going to diverge. They are very different. They're being driven by different populations. Over time, if they come into the same place, they can be different from each other. And then host immunity, we have a very strong immune system that allows us to often clear viruses and can really determine the direction and the trajectory of viral evolution. So I mentioned phylogenetic trees. Let's bring forward a toy tree. If you've seen me speak before, I like to use these toys to try and introduce trees to you and give you an idea of how we actually might interpret them. So we have some dimensions on a tree. It's important to remember that the horizontal dimension is the one that is interpretable. The dimension going up and down, that vertical dimension, is non-interpretable. There's been efforts to add data to that vertical dimension, but in most cases, we don't actually infer any sort of importance from a virus being further up on the page versus being at the bottom of the page. But in fact, it is that horizontal dimension that gives you an ability to understand relationships. That horizontal dimension is often the amount of genetic change. The longer a branch is, the more genetic change has happened on that branch, the more time that has gone between that observation of a virus and its ancestor. This is the amount of change that happens over time. This can be a measurement of substitutions per site or actually units of time. And in this case, I'm saying that 0.5 nucleotide substitutions per site is what that branch length means. So if you have 1,000 base pairs and you see this branch length, you can assume that about 500 of those base pairs have changed over that period of time. This is a tree. All trees have leaves at the ends of branches. These are we call them leaves. They could also be called the taxon or the tips of the phylogenetic trees. Leaves are connected to branches. Branches come together at nodes. Just like if you were looking at an oak tree, as two branches come together, that node is where those branches merge. In this case, where those branches merge, we say that is our putative common ancestor. That is the shared common ancestor of these different observations. And then the roots, often we observe roots as growing, or we observe trees as going from the root up. That's our point of reference. And in this case, by adding a root to a phylogenetic tree, we say that evolution has some directionality and the sequence of events move away from that root. Okay, so the root is the common ancestor of all observations that we have. When viruses are grouped together, we call that a monophyletic clade. And if they are not, if our trait of interest, these green squares, for them to be monophyletic, we have to include some of these blue ancestors. We would say that then that is paraphyletic. That is not monophyletic. Where monophyletic only includes this group of interest. So, for example, if you were interested in, say, identifying a important vaccine escape, if you see all these groups that blue ones are vaccinated, and we get a group of green here that is monophyletic, we would say, "Ooh, we have a monophyletic lineage that has escaped vaccination." That is a different group. Where here we might say strains are repeatedly escaping vaccination. Okay, just to give you an idea of how you might interpret those different tree grouping. To go a little bit further, maybe we have an example here where we have some squares that were from a cold environment and other ones that were from a hot environment. So what we can do is we can actually take those hot ones and put them in a circle or a box. We say these hot ones are all monophyletic, they're a group, and we say all these blue ones, they're monophyletic and they're in a separate group, and they're cold. So the cold ones group together and the hot ones group together, and all of them fit into one box. Maybe they're all bacteria or they're all viruses, but they all have a particular trait. So all these ones that are either in hot environments versus cold environments are related, but the hot ones are distinct from those cold ones. If these are bacteria from deserts around the world, we have deserts that are cold deserts and other ones that are hot deserts. And they're spread all over the world. We can start putting some geographic traits in here to try and interpret this tree as well. And in this case, we would say, wow, these cold adapted organisms are more closely related to each other than ones that are close between. So these bacteria from the Arctic and the Antarctic are more closely related to each other than they are to any of those that are found within North America or in Asia or Africa. So the ones at the polar extremes are related to each other, and the ones in the middle are related to each other. So it gives us an idea about what might be driving the process that's generated this this pattern of spread. And we would say, hey, it looks like it's environmental selection in this case. We add another example. Let's say we have a hospital outbreak of bacteria. We have hospital 1 in green and hospital 2 hospital 2 in green, hospital 1 in blue. Excuse me. So maybe this is an antibiotic resistant outbreak. We want to know whether these outbreaks between these two hospitals are distinct, or if they are possibly related to each other. So the first look, we say, oh well, the green ones, the hospital outbreak the outbreak in hospital 2 is not monophyletic, and we could say, well, these two at the bottom, they fit into a group together. They were likely infected from the same source, but this particular group required some or shares a common source with those two individuals in hospital 1. And in fact, we need to include this third outbreak within hospital 1 to start seeing how those the spread within hospital 2 is not related to these two groups. So we have to start taking a wider view than realizing that the source of the outbreak might be, in this case, related to patient movement between different hospitals, and ultimately it may have come from hospital 1 from some source, but it's moved between different hospitals. And some of those longer branches might say, but we've missed some cases in between there. Okay, so this is how we look at those relationships, and we might try and make some inferences about how we would use a phylogenetic tree to inform our understanding of a disease investigation, and hopefully come up with strategies to mitigate and control that disease. Trees themselves have lots of various shapes. These are all trees that I've made from H3 influenza that were found within humans, which have this, let's see, can you guys see my pointer? People see the pointer? I'm gonna hope you can. Yep, I got some thumbs up here. Great.
Yes,
So seasonal human influenza, H3 viruses, this is typically what the phylogenetic tree looks like. It is a spindly tree. We have many tips that are generated during an epidemic, but very little of that variation actually persists. We believe that that is a result of strong human immune selection, removing most of the variants. And the variants that success are just slightly advanced of our immune system. They're able to escape our immune response. And we end up getting a tree that, if you look at any individual time point, most of these viruses, say in the most recent part of the time, you only have to go back about 1 to 2 years to find a common ancestor. Where if you look at this time point in avian influenza, again, H3 viruses found in wild birds, at this time point here, if we put a line to find a common ancestor, we actually have to go back all the way to the roots, which in this case is about the 1850s. Okay, so human influenza, we only have to go back 1 to 2 years to find a common ancestor, and this is for viruses that are circulating all over the world. With avian influenza H3, we have to go back to about the 1850s to find a common ancestor. We also see a lot of geographic structuring in wild birds, where in human influenza we see we don't see geographic structuring. We see viruses moving between the northern epidemic and the southern epidemic. So during an epidemic in the northern hemisphere, we might see Japan, New York, and London clustering somewhat together, and then would have a phase of South Africa and Australian cities grouping together, and then it would mix, and again, we'd see the northern hemisphere epidemic. But we don't get real strong geographic structuring that persists, indicating that the epidemic is moving from the north to the south every year. When we see avian influenza, and in fact, this top clade is all from North America, and the bottom clade, the middle clade is from Eurasia, and then the bottom clade is an introduction into North America again. So we have this long-term geographic structuring that persists for a really long time. If we look at H3 viruses in domestic birds, we get long branches and an explosion of tips, indicating that when we have an outbreak, we have a rapid spread of virus within that population. We seem to have some geographic structuring at the tip, but these groups are often mixed. So the virus is moving very quickly between farms and locations. And we might not know what that mechanism is. Maybe it's wild bird migration, maybe it's animal trade in there. But we have a way of interpreting this by knowing something about the where these viruses were collected, the time that they were collected, and possibly the relationship with other farms in the region. And it gives us an ability to interpret that tree. And as you continue through your learning and your progress of molecular epidemiology and using molecular epidemiology, you will make lots of these trees. There's a lot of knowledge that has to go into making the trees, but really I'm trying to focus on how you're going to interpret those and trying to extract that information that might be useful for you as epidemiologists. So if the tree the tree shapes themselves are determined by the host ecology and epidemiology, and those actual tree shapes are really important, and they're not always consistent. Is the message that I'm trying to get here, and understanding that when you build a phylogenetic tree, you might get something that looks very different. So if you build one for influenza, seasonal influenza, you'd get one that looks a little bit like the one on the right. If you did one for SARS-CoV-2, early on in the epidemic anyways, you would probably get one that looks a little bit more like avian influenza here in domestic poultry. So the differences there really have to do with host immunity and host ecology, the spread between populations. The tree shape is a product of its historical processes. And when we do get clustering, that where things are phylogenetically monophyletic, that could be due to geography, or it could be due to being a slow-moving pathogen, like something you might see in a hospital, or it could be a localized outbreak where it hasn't spread beyond certain confines. Maybe it's localized just to a elderly home or a care home for individuals that might be immunocompromised. In this example, we have H4 influenza, which is a low pathogenic influenza found in wild birds, and we have this geographic clustering of North American viruses and Eurasian viruses. And that spread is really, that shape of that tree is, and making that big bushy tree, is because viruses take a long time to move between these different locations. So the speed of movement can also influence that shape of tree as well. When we examine that H1 viruses and try to understand the process by which viruses have emerged, when we get a big bushy tree that has suddenly the branch it starts looking different. So at the top we have wild avian viruses. It's pretty bushy. Then we have H1 seasonal H1 viruses started from the 1918 epidemic, and the pattern becomes very ladder-like. And then this is this is a swine influenza at the bottom grouping here. So that swine influenza also has a slightly different pattern as well. Okay. One of the reasons why we can make this type of interpretation is because viruses evolve very rapidly. And when we examine those gene sequences, we line them up, we build the phylogenetic tree, we're seeing the signal of that mutation, migration, selection, and genetic drift that is present within that population of gene sequences. We can make inferences of past events based on the branch patterns and those branch lengths. So long branch lengths might say, hey, we have a period of missing data or a period of rapid evolution. If we have a lot of very short branches and rapid spread, we might say that we're having rapid evolution, or increased number of infected individuals, a rapidly increasing spread within a population. This is actually one of the characteristics that we use to identify a variant of concern, that if we have a lot of short branches and a rapid spread, meaning rapid increase in incidence, both collected from individuals, but also modeled on the phylogenetic tree, we would say, hey, this is probably a variant of concern. And then that triggers a different set of observation criteria for SARS-CoV-2, for example. So the evolution seems to occur actually on the same time scale as the ecological and the epidemiological spread and interactions that occur, and that's very different from, say, bacteria or mammals. So where viruses, these things happen on the same time scale. With bacteria, it's slightly more delayed. It often can be interpreted down to months and years. And then with mammals, for example, we're talking thousands and millions of years. Especially if we start going between species, we might be going millions of years. So capturing the interpretation is still it's still capturing the signal of those different evolutionary processes, but the interaction with ecology happens on different scales. Where viruses, it's on the same scale as we're observing the data. Capturing signals of geographic movement is probably some of the most important inferences that you will make in that you can make very rapidly from building a phylogenetic tree and without having to generate really complex geographic models. Of course, the geographic models help you have increased confidence in your inference, but just by building that phylogenetic tree, we often see that influence viruses, in this case being high-path avian influenza, will group together based on geographic region. In 1997 to 2003, high-path avian influenza was restricted to southern China, and it was often observed in imported birds and into Hong Kong. But then in about 2003, it suddenly emerged in Southeast Asia, in Vietnam, Thailand, and Malaysia, and then a separate introduction into Indonesia. This is a project that I worked on early in my career as a molecular epidemiologist, and I was living in Hong Kong, and we had many samples that we had collected throughout Southeast Asia, throughout southern China, through the market system and through the bird production system. So we had a whole bunch of different species of poultry, chickens, pheasants, duck, goose, quail, chukka, different poultry animals that were commercially sold. And we had collected high-path avian influenza from many of these different species, and we knew where they were found throughout southern China. Luckily, that many of these viruses, because they do evolve so rapidly, we could see that signal of geographic clustering within the tree. So we would often have viruses from Yunnan grouping together, viruses in the Hunan grouping together, viruses from Shantou grouping together, and then this group at the bottom here are the viruses that were found from Vietnam, Thailand, and Malaysia, ones that got into Southeast Asia. When we included all the viruses that we found throughout southern China, we had all these viruses from Yunnan province that shared a common ancestor with this group from Vietnam, Thailand, and Malaysia. So this group here was monophyletic, that if we included these individuals from Yunnan province. If you're familiar with the geography of southern China, this is Yunnan province right here in the western part of southern China, and it shares a land border with Vietnam and Laos here, and connects itself with the rest of Southeast Asia through those land borders. So this provided some evidence that the outbreak within Southeast Asia was connected to these viruses that were circulating in the poultry production system within Yunnan province. And then subsequently, we received information that smuggled duck eggs and smuggled chickens were positive. They were being smuggled over from Yunnan into across the Vietnamese border here. They were positive for high-path avian influenza. And it looks like that introduction actually sparked the outbreak that spread all the way across through Southeast Asia. Indonesia, on the other hand, was separately monophyletic, and it was connected with viruses found in Hunan province, which is it does not have a clear land border, and the mechanism of introduction is still unclear. We don't know if it got back into wild birds at that point, and wild birds made that migration, or if it was birds that were positive for high path that were kept on fishing vessels that had made the journey between these different locations. So we don't know how it actually spread, but we were able to look at or get some information on that epidemiological spread and the drivers of spread throughout the region from building that phylogenetic tree and knowing where viruses came from and how they clustered on that tree. So in this case, that Vietnam, Thailand, Malaysia was monophyletic, and it shared a common ancestor with these viruses that were restricted to Yunnan province. I think that was very strong evidence. And then we found additional evidence through investigations of what was being smuggled over that border. We also detect signals of interspecies transmission. This is the emergence of pandemic H1N1 virus. These ones at the bottom in red were the earliest H1N1 2009 pandemic viruses found from individuals in California and Texas. These two groups were not epidemiologically linked, but they were monophyletic, so they were all being infected from the same virus, but these groups were not linked in a clear way, so we don't know how they got infected. When we started looking at viruses that were circulating in other populations, we quickly found that we had common ancestors with viruses that were circulating in Hong Kong swine a few years earlier, and we were able to work out how that entire virus that caused the pandemic was generated through spillover with swine viruses. We do have a pretty interesting phenomenon in influenza in particular, which is called reassortment. It means that viruses mix together very frequently, and this is how pandemics can be generated. But we also have recombination and lateral gene flow, which is a bigger issue in bacterial pathogens. So if you are looking at antimicrobial resistant outbreaks, you have to be aware of recombination. In viruses, it's when two different viruses infect the same cell, and we have the exchange of viruses of gene segments either through reassortment or through copy choice slippage that allows recombination to happen. Recombination in viruses happens at a much lower frequency than reassortment does. But when recombination does happen, it can give rise to things like SARS-CoV-2. So it doesn't happen as often, but it does happen. Reassortment in influenza happens a lot, and this is a major confounder in trying to control diseases and stop pandemics, but it also causes a lot of problem in creating phylogenetic trees. So I wanted you to be aware that if you produce a phylogenetic tree with one target gene sequence, and then you get the whole genome and you get a completely different pattern, it could be because of reassortment. And we call this gene tree discordance. And when we have these different patterns from different trees, this could be something that gives you wrong inferences. So hopefully through efforts like the Pathogen Genomics Center of Excellence and interactions with other experts, we will be able to provide guidance and through CDC, provide guidance on what gene targets you should be looking at for molecular epi. But I do want you to be aware that if you don't consider reassortment and recombination, you could get wrong inferences, and that will affect your control efforts. Okay, so just put that out there as a warning. Hopefully we'll devote some time during the workshop that we have scheduled for later in the year to actually dive into how we have recognized what reassortment and recombination is. With influenza, the nice thing is that we can use these 8 different genes that have 8 different evolutionary histories, so they have 8 different trees, to understand the interaction of different different populations and viral gene flow between different populations. So for high path avian influenza, we were able to understand how different variants were being generated through viral flow from wild and domestic animals, and geographic spread, and then with with the emergence of pandemic H1N1 2009 viruses, we're able to see that over a period of 30 years, we had viruses that moved between avian hosts in the top in the green to the middle swine populations, and even going from seasonal influenza back into swine. And as these viruses bounced around, eventually coming back into humans and causing the 2009 H1N1 pandemic. Okay, so just to, I think, try to round things out, my definition of a phylogenetic tree is that this is a hypothesis of common ancestry based on similarities and differences. When we use genetic data, our measurements of similarities and differences are genetic, and that's how we start pulling in information on, we build the tree with the genetic difference, and then we can start understanding how geography or epidemiology might affect those patterns that we're seeing. Evolutionary processes give rise to diversity, and branching patterns and branch lengths are the result of evolutionary processes. So the tree patterns and the tree shape, so the grouping, the clustering, and the tree shape is a product of natural selection, geographic movement, mutations, and genetic drift. That phylogenetic tree gives us a framework to interpret how epidemiology and ecological processes can be observed on that same tree and same time scale. Ecological, epidemiological, and evolutionary inferences for viruses occur on the same time scale. When we start doing model-based inferences of phylogenetic trees, we can provide estimates of that evolutionary history, showing when we have an ecological bottleneck, that a virus has jumped to a different species and we've had a loss of diversity. When we have had a change in the transmission patterns, when we're looking at the population genetics for viruses, we are looking at how many people are actually infected. So if we have evidence that there is an exponential growth in the viral population genetics, that would say that we're going through a period of exponential transmission as well. And capturing those measurements of host shifts, which I mentioned as well. All of these things impact the genetic diversity that we see, and we need to start looking at these things on the same time scale, especially for viruses. Viral phylogenomics, this field that was introduced in 2004, is a study of how epidemiological, immunological, and evolutionary processes act and shape and interact that phylogenetic tree. All these things come together, and this is often the field this is often what molecular epidemiology that learns from phylogenetic trees is called. As people here, if you're working in the influenza branch and you're getting data and you start building phylogenetic trees and trying to understand how that epidemic within Georgia, is it circulating locally or has it been introduced into Georgia, that is ultimately the field of phylodynamics. Without going into the mathematics of different models and different modeling that's been done since 2004, which I will say has been a phenomenal amount in trying to unify ecology and epidemiology, just by looking at the shape of the tree without doing these other computationally challenging and advanced mathematical models and statistical tests. Without doing any of those, you'll still be able to make useful and informative inferences to guide your response. With the improvements in genetic sequencing, you should be getting sequences ongoing as an epidemic happens. This is the goal for the future epidemics, that as samples come in, you'll be able to get viral sequences, and you'll be able to produce these phylogenetic trees and rapidly determine whether you're actually having an outbreak within a care facility, or if outbreaks within hospitals might be connected through these patterns of common ancestry, indicating these apparently separate hospitals actually are being infected from the same source. This allows you to guide your investigations and hopefully enhance what you've already been doing, and what you've been doing incredibly well as well through your practice of epidemiology. So I know I covered a lot of stuff. I believe that's my last slide. That is my last slide. I am very happy to take questions